Jason Bubier, do I pronounce that right?
Yeah.
I don't believe you.
No, it's the name sounds French, but my DNA is all English, so it just didn't go together.
Okay. All right, good. That's fine. I appreciate that. He's a senior research scientist at the Jackson Laboratory, and his work sits at the intersection of behavior and public health, and his profile reflects a career shaped by curiosity about why individuals respond so differently to addictive substances. His narrative is one of scientific persistence, following complex biological questions across disciplines to uncover insights that could ultimately save lives. Jason's science-adjacent popular culture item is near and dear to my heart because it is the Star Wars universe.
Thank you. And my 17-year-old son said I could not show up wearing my Luke Skywalker pajamas. So the socks had to be it. All right, I'm here to talk to you about something that, 2,000 years ago, Hippocrates said is that all disease begins in the gut. So it's been a few years since he said that, and now technology has improved, and we've done a lot of new cool things with science, including the Human Genome Project in 2003, the 1,000 Genomes Project in 2015, and one of the things we found out is that these minifigs representing the human population is that they're all very different, but actually we're 99.9% similar. So this was kind of startling to see, especially when we thought, okay, we'll find the gene for everything. 20,000 genes, we'll find a gene for the blue hair. But there's actually this missing heritability. Previous work using twin studies, like identical and a non-identical twin studies, and looking for the heritability or the inheritance of different traits, was suggesting, oh yeah, schizophrenia, it's going to be 50% heritable, and it should be a single gene, and it's not. It's hundreds of genes. And so these QR codes should take you to these different articles to learn more about the missing heritability. So what I want to do is tell you about it, that the second part, so this didn't come through right, that's supposed to be over there. The Human Microbiome Project was launched in completed in 2016. And what we found out there is that actually who's running this ship is as much microbes as it is humans. And when you look at it by bacterial weight, the human bacterial 70 kilograms to bacteria, 0.2 kilograms. When we look at the cell level, it's about 1-to-1 or 1-to-1.3. So there's, more cells that are not you than there are cells that are you. And then when you look at the gene level, and people have been talking about this in the genes that are expressed in the 20,000 and 30,000 and that are in the plankton, the gut bacterial composite toolkit or list of parts that could be found is 2 million genes. So that's 100 times more than we have. And these commensals or symbionts, they live in our gut. I'm mostly focused on the ones in the gut, but there's a whole microbiome on the skin. There's microbiomes all over the body. But the gut microbiome, perhaps the source of disease, is what I'm interested in. And this is supposed to be a larger graphic. And the gut-brain axis is an emerging field of study in which people are learning that what's going on in the gut is actually affecting the brain. 50% of your serotonin, a neurotransmitter, 90% of your dopamine, another neurotransmitter, are created in the gut. There are microbes in your gut, such as Tertiobacter, that have transporters in their membrane that are made to transport your serotonin. So there's a relationship here. I mean, these bugs have been around with us as we've been evolving through the years, and they've found ways to survive, including feeding on our serotonin. Another thing that's found in the gut is as GABA through the process of glutamate. So the summary here is that the healthy brain, in a normal state, you have a good balance of your microbiome in the diversity of bugs that you have and what they are doing. But when you are in a diseased state or a pathological state, you end up with possibly a dysbiosis, a dysregulation of your flora. Things get out of whack. You can end up with a leaky gut, meaning that the cells in the gut are leaking, and components such as polysaccharides can leak out and stimulate your immune system, get neuroinflammation. Bad things can happen. So the disordered brain can occur because of what's going on in your gut. Which brings me to the one thing that's going to talk about what I actually do, and that is this particular paper. Microbial glutamate metabolism predicts intravenous cocaine self-administration and diversity outbred in mice. So I work at Jackson Lab. I give mice cocaine, or actually I let them give themselves cocaine. It's an intravenous volitional approach. They tap on a lever, and some mice love it. Some mice, I can't get addicted. And you know what? The microbial glutamate metabolism pathways, some of those 2 million different pathways that I was talking about, are enriched in those mice that do not become addicted, that I cannot get addicted. In addition, it's a correlation at this point, but there's also genetic causality, because as I showed in those different Lego pictures, the red hat, for example, could be your ability to be cultured by a bacteria. So certainly you may carry a gene that allows you to be cultured, to be inhabited by a certain bug. So again, you have to think about this arms race over time. And so just to wrap it up, my biggest point is that the gut and microbiome is a new area where we could make targeted therapeutics for things such as substance use disorder, and let food be thy medicine and medicine be thy food.
Kind of a two-part question, completely unrelated, but I didn't want to take up too much time. How does the vagus nerve play into all the things that you do? And then how do you see GLP-1s playing into what you do?
Am I at a science conference here? Very good questions. And I have to say that the vagus nerve is part of the part of the gut-brain axis, a major signal transference between the gut and the brain. In lots of animal studies, they go in and they sever the vagus as a way to see if that is the pathway. So there's many mechanisms of communication between the gut and the brain, and the vagus is one such mechanism. And GLP-1s are showing great potential for, at least in some studies, for alcohol consumption, having to do with, satiety and gastric motility. And microbes are in your gut, and they, if you inhibit
through opioids actually cause constipation. It can affect the gut microbiome as well. A single dose of fentanyl
can be detected for 14 days later because your gut is really messed up by that. So yes, GLP-1s, stay tuned. I mean, they're going to be marketed for that next too.
You mentioned identical twins as part of what you've been aware of. Have you been able to get identical mice and addict one but not the other? And also, are you familiar with E. Fuller Torrey and his research on schizophrenic and non-schizophrenic identical twins?
The second part, no. The first part, yes, there are, which is amazing. There are strains of mice that are genetically identical clones. For example, Black 6J is our common strain, our John Doe mouse. Everybody's supposed to be genetically identical, clonal. I can only get 50% of those mice to become addicted to cocaine, even though they're identical at all levels. So it's not nature or nurture, it's both. It's genes by environment interaction. So even in clonal animals, we see variation.
Hi, I saw depression on your, your slide there, and I was curious, is this work in any way leading toward, somebody has depression and you can test their gut to see what kind of medicine would work best for them?
I think that's definitely in the future, is that we can test their gut and see either what type of diet modification might be good, what kind of bug inoculation, or a drug that might have been made by a bug that you could feed to somebody, like a bile acid or an amino acid or something. So yes, it's definitely, and depression and anxiety are comorbid with substance use disorder. We never know which is causal, so we are always studying those behaviors as well. Whenever we study addiction, those same mice, we put through studies where we study their ability to see how depressed they are, how anxious they are. So we're measuring a variety of behaviors before we get them addicted, and then to relate them to one another.
Thank you.
I'm wondering if you also have studied acetylcholine, and if acetylcholine is also in your gut, and do anticholinergics impact that?
Yeah. So I just want to say that I'm a geneticist and a biologist, not a neuroscientist, so no.
Hi, it's very similar to her question, but does this mean that you think we're maybe close to a pill or juice you can drink that'll prevent addiction?
I don't know if we're close, but we're beginning to understand that this is a new doorway that we could be walking through, and that this is a new frame of thought, and we don't need to be just focusing on the things that we're thinking of currently, that this could be another way to target because of the historical coevolution of the bugs and our guts together. Some people posit that addiction is actually it would have been a trait that would be actually advantageous during evolution. So if you're trying to obsessively find something or become obsessed with something, that could be a trait that is encouraging survival, but it's maladaptive at this point when in our society.
Are there foods that you find are helpful and harmful?
I haven't done any of the interventions, so one of my favorite parts of working at the Jackson Lab is the controlled environment. I can, unlike humans with mice, I can control exactly what they eat, when they eat it, how much they eat, what time of day it is, and how great their healthcare is. But with a human situation, I can't do that.